Hello guys and welcome back to Excel Sure Academy. So in the previous lecture we were discussing about tRNA, right? So just to summarize, so we have talked about the 2D structure of tRNA and also about the 3D structure, right? So if you remember, we have discussed this slide also. So here in which you can see that how from 2D to 3D it is looking, right? And we have discussed that this is this arm where it is 5-CCA 3 dash. The sequence is single stranded and it is also known as the acceptor arm of tRNA, correct? Then we have got the anticodon region. Then of course we have here the dihydrouridine group. Here it is pseudouridine group and the variable loop of your tRNA. Okay. Now, same slide, just to summarize. So, amino-acid tRNA synthetases charges tRNAs in 2 steps. So, first one is the adenylation of the amino acid. So, ATP is required. So, what you can see, CCOO dash, that is your carboxyl group is attacking over here, right? Then there is a release of the pyrophosphate. Then pyrophosphatase is going to give you 2 inorganic pi, and that energy is sufficient for this adenylated amino acid to get loaded onto the tRNA. Okay, so then this 3-OH is there attached over here and it gets attached to the amino acid by releasing AMP. Okay, now we also discussed that when it is 3-OH, it is which class of which class of tRNA synthetase? Class 1 or class 2? Do you remember the table? So, are you able to remember? तन्वी याद है? अगर 3-OH है तो कौन सा क्लास हुआ tRNA सिंथेसिस का?
मैम 2 सेकेंड.
यस, और अगर 2-OH हुआ तो क्लास 1 हो जाएगा. ओके, गुड. This is the same figure which we have discussed earlier. So here you can see that the amino acid, ATP and the tRNA, for example, in this case it is methionine, it has got the binding site at the tRNA synthetase, amino acid tRNA synthetase. Okay, then later on what you see is that first your methionine or the amino acid gets adenylated with the help of the ATP, releasing your 2pi. And then later, once this adenylated amino acid gets charged over the tRNA, it releases AMP. So at the end, what we get is the charged tRNA with the respective or the cognate amino acid. Okay. Now, the tRNA synthetases recognizes unique structural features of the cognate tRNAs. Cognate tRNAs मतलब क्या हुआ? जिस कोडॉन का जो tRNA है वही particular amino acid, ठीक है? So an amino acid tRNA synthetases, it faces 2 important challenges. So let's see what are those 2 important challenges. So they must recognize the correct set of tRNAs for a particular amino acid, and they must charge all of these iso-accepting tRNAs with the correct amino acid. So already we have seen, right? Ki serine hai, and suppose let us say that it has got 6 codons, right? So this amino acyl tRNA synthetase for tRNA, अब इसके पास तो 6 एंटीकोडॉन भी होंगे which are different and it has to get charged only by serine. तो ये करने के लिए, okay, so iso accepting tRNAs means having different anticodons but binding or getting attached to the same amino acid. Okay, so how the amino acyl tRNA synthetase is making sure or performing this with high fidelity. So उसके लिए भी हम देखने वाले हैं. So already we have mentioned that it is all because of those anticodons and acceptor arm इस पे based है जो आपका amino acyl tRNA synthetases understands. It reads the anticodon present on the tRNA so that the correct amino acid is getting attached. Okay. So there were many genetic and biochemical X-ray crystallographic studies which were done, which gave the evidence indicating that the specificity determinants are clustered at 2 distant sites on the molecule. तो पहला हो गया आपका acceptor arm and दूसरा हो गया आपका anticodon loop. Okay. So many other, see when you talk about genetic biochemical X-ray, so what we do, we try to make mutants. Genetic मतलब क्या? We are making mutants and we try to see whether it is possible to change the anticodon and still it is coding or still it is getting charged with the same amino acid or the other different amino acids. So इस तरह के experiments करके पता किया है कि these are the 2 important regions. So one is the acceptor arm and the another one is the anticodon. Okay. So the acceptor stem is an especially important determinant for the specificity for tRNA synthetase recognition. So guys, so this point is clear, right? कि हमारा anticodon होना और acceptor end होना. Okay, these are two important features by which tRNA synthetase is able to determine and choose a correct amino acid for that particular tRNA. ठीक है? In some cases, changing a single base in the acceptor arm, which is also known as discriminator base, is sufficient to convert the recognition specificity of a tRNA from one synthetase to another. Okay, ये example है जहाँ पे इन्होंने क्या किया है, nucleotide sequence change किया है, ठीक है? Okay, now the challenge faced by amino acid tRNA synthetase in selecting the correct amino acid is perhaps even more daunting. क्यों? Because we know that in case of amino acids, there are sometimes they are similar in shape or size or because of the chemical groups, right? अब जैसे कि glycine है, it is so small amino, it's a small amino acid, correct? So it is possible that it can get fit into the amino acid tRNA synthetase of another amino acid क्योंकि वो छोटा है. Are you able to follow, guys? Able to follow? अभी हम जो डिस्कस कर रहे हैं, so अभी इसमें क्या बोल रहे हैं कि how the tRNA synthetase, the enzyme, okay, is able to differentiate between the amino acids when they have got this similarity. For example, if you have seen the structure of phenylalanine and tyrosine, इन दोनों के साइड चेन ग्रुप में किस ग्रुप का अंतर आता है? If you have seen the structure, how much difference is there? Yes.
There is a CH3 group extra in phenylalanine.
No. There is no CH3 group. CH3 group nahi hai. So these are your aromatic amino acids, correct? So see, I'll draw for you then. Okay, so it's like this. So you have got the basic structure जहाँ पे आप पहले ही से NH2 लिख दो, COOH लिख दो और H लिख दो. Now to the side chain, ठीक है? Side chain CH2 and then you have got the ring. Okay, so this is your phenylalanine का structure. Now, in case of tyrosine, it is very much similar. Last में सिर्फ एक OH group आता है, ठीक है? So if you see the structures are very much similar, it just that इतना सा आपका एक alcohol functional group is present. Okay, so there might be a chance जहाँ पे tRNA synthetase can make a mistake of taking tyrosine instead of phenylalanine or vice versa. तो ऐसा भी हो सकता है, right? So the question is how this high accuracy is being achieved. So for example, the amino acids cysteine and tryptophan, they differ substantially in size, shape and chemical groups. So अगर आपके पास amino acids हैं, so definitely any enzyme can differentiate. Okay, Akshay, able to follow like what we are discussing right now?
Yes, ma'am.
Okay, so if the difference is there between cysteine and tryptophan, it is very easy for an enzyme to differentiate. Okay, problem कहाँ आती है? जहाँ पे you have got these similar groups. For example, ये आपका tyrosine हुआ, same thing, NH2, COH, CH, CH2, phenyl group is there, then additional OH is present in the case of tyrosine, whereas in phenylalanine it is not there. Okay, so even in the case of the similar looking amino acids like tyrosine and phenylalanine, the opportunity for forming a strong and energetically favorable hydrogen bond with the hydroxyl moiety. So agar aap dekho, look at the structure, phenylalanine ka in this case. So who has got more hydrogen bonding opportunity? Of course, the water loving hydroxyl group. Okay, so it can form it can form very nicely and easily hydrogen bonds with the enzymes. Okay, whereas in the case of phenylalanine, it is not possible. So this is how the enzymes are going to differentiate. ठीक है? So for example, अगर मैं लेती हूँ tyrosyl tRNA synthetase, okay, what does it mean? So this tRNA synthetase is going to attach amino acid tyrosin to the tRNA. इसका ये मतलब हुआ, okay? Now, so this tRNA synthetase, if tyrosin is coming, then it is going to make additional contact with the OH of the amino acid. Okay. Now, if I had phenylalanine tRNA synthetase, okay. Now, if this enzyme is present and tyrosin is coming, so what will happen? Because of the presence of this OH group, there will be a steric clash. Yes? वो एडिशनल ग्रुप है वहां पे प्रेजेंट है, जिसकी वजह से क्या होगा, द phenylalanine tRNA synthetase does not have space for that additional OH group, जिसकी वजह से क्या होगा, because of the steric hindrance, the amino acid won't be able to sit onto the tRNA synthetase of phenylalanine. Only phenylalanine can come. Okay? अगर आपको कोई ये बोले तो ठीक है, steric clash हुआ. What about glycine? Glycine is such a small amino acid. It can come and sit with any tRNA synthetase. Correct? अगर उसके पास इतनी जगह है, हमारा glycine छोटा सा, it can come and sit over there. But again, the glycine won't be able to sit there properly. It will be loosely bound. Correct? Because it won't be able to make the interactions with the binding pocket of the tRNA synthetase. Hence, it will again leave the binding pocket and that space will be occupied by the phenylalanine amino acids only. Yes, Tanvi, are you able to follow जो भी हमने discuss किया?
Yes, ma'am.
Okay. Akshay, what about you? Followed? Shall we move ahead?
Yes, ma'am.
Okay, good. Now the problem comes when between, for example, isoleucine valine, which differ only by a single methylene group. So same explanation जैसे भी मैंने OH के लिए दिया, same for the methylene group also. ठीक है? So tRNA synthetase of isoleucine will have additional space to interact with the methyl group, whereas valent tRNA synthetase won't have. अगर यहाँ पे vice versa होगा, again just because of the space limitation, ठीक है? Valent tRNA synthetase and over there isoleucine amino acid अगर आता है, just because of the additional group, it won't be able to fit into the binding pocket of the valent tRNA synthetase and vice versa. Okay, simple. So this is how it is maintaining the high fidelity in charging the tRNA. Okay, so this is another example वही जो भी हमने discuss किया. So we'll just read once. For example, example 2. So पहले हमने tyrosin and phenylalanine का लिया, अभी हम लेते हैं valine and isoleucine का. So valine tRNA synthetase can sterically exclude isoleucine from its catalytic pocket. Why? Because of that additional methyl group present. So it is larger in size as compared to the valine. Okay. And in contrast, valine should slip easily into the catalytic pocket of the isoleucyl tRNA synthetase. But now, although both amino acids will fit into the isoleucyl tRNA synthetase amino acid binding site, interactions which is made with the methylene group, जो isoleucine के पास है, will have more, will provide an additional -2 to -3 kilocalories per mole of free energy. ठीक है? So this additional binding will make sure that this tRNA synthetase is correct for this particular amino acid, and hence the charging of the tRNA will take place. Okay, clear? Is this sentence is clear? अगर एक बार बोल दो कि then only I can go ahead, because these are very important to understand. Yes, guys, able to follow? This sentence is clear? Yeah, okay. So even this relatively small difference in free energy will make binding to isoleucine 100-fold more likely than binding to the valine. So this 100-fold more likely is making sure that this is the correct attachment. Okay, and the error rate will be decreased. So that's how they are achieving the level or high level of fidelity, यानी कि कितना सही amino acid attach हुआ है to the tRNA. Okay. Now, some amino acid tRNA synthesis, they use editing pocket to charge tRNA with the high accuracy. So already we have discussed like what are the mechanism by which the amino acid tRNA synthetases are making sure that the correct amino acid has been charged. It's first it is checking its catalytic pocket जहाँ पे आपका adenylation हो रहा है, ठीक है? So as we have seen in the figure earlier, let us say that this is the pocket. So here your amino acid should come and sit. अब ATP तो सभी का common होगा, like all amino acid tRNA synthetases will have same binding pocket for the ATP because ATP आपका change नहीं होगा. The only difference will come in the case of the amino acid, ठीक है? And the tRNA depending on the anticodons. Okay. So first is the checking of your catalytic pocket where you are going to have the adenylated amino acid. Now, so whether they are able to fit into this editing pocket or not, where it is hydrolyzed and released as free valine and AMP later on. Okay. So एक आपका ये तरीका हो गया, and this is how it is making sure that it is able to charge the correct amino acid. तो आपका first checking वहां पर ही हो जाता है. I have the figure also. Yeah. Look it over here. I don't know how much will be clear by this. So we have got this. See, you can see that यहाँ पे tRNA है. Okay, it has bound with the wrong amino acid. So it is making sure that okay, wrong amino acid हो गया, that's why it is not able to fit properly. So they have got the editing side. So editing side में क्या होगा? The incorrect amino acid will be removed. Okay, and then later again the new amino acid, the correct amino acid will be brought into the binding pocket so that the correct amino acid is charged onto the tRNA. Okay, clear now? So this is called hydrolytic editing. Just ये अभी देखो analogy के लिए इन्होंने यहाँ पे DNA polymerase का दिया. If you remember in our previous lectures when we started about the DNA polymerase, so we know that जब भी DNA polymerase में प्रूफ रीडिंग मैकेनिज्म होता था, जैसे रॉन्ग न्यूक्लियोटाइड इंसर्ट हो गया तो क्या होगा, जिसकी वजह से द बेस पेयरिंग जो रहेगा प्रॉपर नहीं रहेगा, करेक्ट, एंड देयर विल बी सम ट्विस्ट एंड टर्न और बेंड इन द स्ट्रक्चर. So that will make sure or it will activate your 3-5' exonuclease activity of your polymerase. So जैसे वो activate होगा, this bend will be taken and at this 3-5' exonuclease activity of the DNA polymerase will remove the incorrect nucleotide and again it will resume. The moment incorrect nucleotide is removed, 3-5' exonuclease activity of the DNA polymerase decreases और उसका 5-3' polymerase activity increase हो जाता है. Same way over here also. So जैसे ही अगर by chance incorrect amino acid अगर आपका attach हो गया है, so it will activate its editing side, hydrolytic editing side, which will make sure that the tRNA is attached with the correct amino acid only. ठीक है?
Yes, ma'am.
Okay. Now let's talk about the अभी देखो धीरे धीरे. So first we talked in detail about the mRNA, अभी tRNA हो गया. Now we have to jump to the ribosome, and after that only we can start with the translation initiation. And once we start, it's good. I mean, then it is easy to grasp because already you know how the ribosome, tRNA and mRNA is है. अभी हमें क्या करना होगा translation के time पे, इन just तीनों को साथ में लाना है with the addition of another 50 proteins which we need to remember. Okay, don't worry. So the ribosome is unable to discriminate between the correct and incorrect charged tRNA. So इसलिए see अभी throughout the translation, if you see, the main responsibility comes on the amino acid tRNA synthetase. ठीक है, अगर उसने correct tRNA and correct amino acid रखा है, then well good, you're going to get a good polypeptide chain, because ribosome won't understand whether the wrong tRNA, wrong amino acid is coming or not, because उसको नहीं समझ में आएगा. They have got the space only to bring the tRNA, make a codon anticodon base pairing, and जो भी amino acid है, it will be transferred to the peptidyl transferase sorry, to the peptidyl region where it is going to form the peptide bonds. That's all. उसको नहीं समझता. Ribosome doesn't understand कि it's a wrong charge tRNA. Okay. So इसलिए इसलिए it is important that the tRNA synthetase make sure that all the amino acids are charged with its cognate tRNA. Okay. So in other words, the ribosome blindly accepts any charged tRNA that shows a proper codon anticodon interactions. अभी क्या हो सकता है, इसमें प्रॉब्लम कैसे आती है? For example, like how they have done and how they understood, like they carried out the experiments. So what they have done was, so there are many amino acids like which can be aminated or which can be oxidated to form different other amino acids. So उससे क्या होगा, आपने कोडॉन तो चेंज नहीं किया, but whatever the charge amino acid was there, that got changed. Now the ribosome is not able to understand. So depending on the codon, it is going to decode at the mRNA codon. It is going to give that particular amino acid. Okay. So I guess I have the example that will be easy to show. Okay. Yeah. So for example, I have this. So we have got the cysteine, tRNA cysteine. Now, guys, you need to understand over here, like the nomenclature, how we give for tRNA, first of all. Okay. So maybe you can take this example also. So when it is written as cysteine, tRNA cis. So this, it is saying that what amino acid it is carrying. Okay. Now this tRNA cis means it is a tRNA having anticodon for cysteine amino acid. Yes, able to follow? अभी टेक्स्ट बुक में भी आपको ऐसा मिलेगा. So what does it mean? This is the tRNA coding for cysteine and it is charged with amino acid cysteine. Clear? Now there are few examples by by the genetic and the biochemical reactions. Many scientists have changed what they have done. So they have done over here cysteine tRNA cis was there, chemical reduction. So when chemical reduction was done, what had happened over here? So they have removed this thing. Okay, the sulfhydryl group was removed. Now what we got was alanine. Can you see the difference? S, yeah, SH है, the sulfhydryl group is there. यहाँ पे ये H है. So the sulfur group was removed. So what had happened? The amino acid got converted from cysteine to alanine, but the tRNA is still coding for your cysteine. Okay, it has got the anticodon for cysteine. Now in the mRNA, the codons which is meant for cysteine, it is going to base pair with that. But in the polypeptide chain, it is going to insert your alanine. Okay, so this experiment proved that it is the ribosome who does not understand the difference between the wrong and the right charge tRNAs. Okay, clear? Should I repeat? Akshaya, Tanvi, it is important. Okay, agar clear hai, then only we'll go ahead. Or should I repeat? So that's why the high responsibility comes on the amino acid tRNA synthetase to make sure the correct amino acids are charged onto the tRNAs. Clear? Yes, Tanvi, is it clear? Akshay, you?
Yes, ma'am.
Okay. Now about the ribosome. So that was all about the tRNAs. Now we are going to discuss about the ribosome. So in this also many things are there. So the ribosome, so as you know, it is a macromolecular machine that directs the synthesis of the proteins, correct? Now, the machinery for polymerizing amino acid is composed of at least 3 RNA molecules and more than 50 different proteins. And you can see that the size of the ribosome, it's so huge, right? It is in mega. Kilodalton hota hai, ye mega dalton. So it's very heavy. And if you know the structures of the ribosome, like abhi hame pura crystallography se pura structure mila hai of the ribosome, and since we got the complete structure of the ribosome, the people were also nominated, not nominated, they actually won the Nobel Prize for this. If you have heard of it, ribosome structure jisne kiya tha. So they were, Ada Yonath were there, Thomas Cheese were there, and Venkat, kya naam tha? Venkat Radhakrishnan. Okay, so these 3 people, they got the entire ribosome ka structure. Now, why it was difficult? Because ribosome is made up of RNA and plus proteins. Okay, so कैसा होता है, कभी कभी RNA crystallize हो जाता है, प्रोटीन नहीं होता है, प्रोटीन होता है तो RNA होता है. And it was easy because it was difficult because it had so many components. So each, if they are talking about 50 different proteins, so 50 different proteins can be crystallized separately. Okay, and it can be studied, but getting the entire ribosome का crystal was the task. Okay, तो पूरा आपका जो 30S and 50S जो हमारा सब यूनिट रहता है टोटल राइबोसोम स्ट्रक्चर, दिस इज व्हाट दे गॉट क्रिस्टलाइज. सेपरेट तो था हमारे पास, and depending on the structure it was assumed, okay, this might be the 3D structure of the ribosome, but now we have there is the availability of the 3D structure of the entire ribosome. Okay, so that's why they have won the Nobel Prize. Okay, so compared to the speed of the DNA replication, where it adds around 200 to 1000 nucleotides per second, amino acids around 2 to 20 है per second की हम बात कर रहे हैं. ठीक है, so you can see, of course, the translation machinery is slower as compared to the DNA replication, and you can understand also why. DNA replication means it has got such a long, huge chromosomes to be replicated, right? And proteins are few as compared to the DNA, and it has to maintain the fidelity also कि वो wrong amino acids incorporate ना कर दे. So around 2 to 20 amino acids are added per second, but you can see that in a minute per second अगर 20 amino acids they are adding in 60 seconds, that is your 1 minute, right? Around 1200 amino acids are added, and this is sufficient to make a one polypeptide chain. Okay, so again, it is quite good velocity of the polypeptide chain synthesis. Now, in prokaryotes, the transcription machinery and the translation machinery are in the same compartment, and that's why it can get coupled. So, couple का मतलब क्या हुआ? So, as coupling, okay, so this is also called transcriptional translation coupling. So, what happens over here is that as your 5' mRNA is coming out, okay, the moment it is out, to that your 30S subunit will come and start scanning, and then 50S subunit will come, the entire ribosome, and they can start forming the polypeptide chain. As you can see in the figure also, it's not that only one ribosome, so multiple ribosome can come together and they can start making multiple proteins. So they are the same protein but multiple copies. So what does it mean? So it means that अगर ऐसा कोई scenario है, so that particular protein is overexpressed. Clear? So here your transcription and the translation is coupled. So जैसे ही आपका transcription खत्म होता जाएगा, side में आपका protein भी बनता रहेगा. So the gap between the transcription and translation is negligible. It's hardly few seconds का gap. Okay, is it clear to you, both of you?
Yes, ma'am.
Yeah, simple topic, and these are the words which people ask in the exam. I'll tell you what are those. So the typical prokaryotic rate of translation of 20 amino acids per second. It means translation of 60 nucleotides, that is 20 codons of mRNA per second. So which is similar to the rate of 50 to 100 nucleotides per second synthesized by RNA polymerase. इसका मतलब almost transcription and translation same rate में चलता है आपका, ठीक है? Now coming to the eukaryotes. So it is different. So we know that in the case of the eukaryotes, we have got the compartmentalization of the cell. So आपका transcription nucleus में होता है, translation आपका cytoplasm में होता है. Now because there is not possible to couple the transcription and translation in the case of the eukaryotes, what happens? ये आराम से होता है फिर. So the eukaryotic translation speed is less as compared to the bacteria, and it makes sense also, right? क्योंकि bacteria को 20-30 मिनट में replicate होना होता है, divide होना होता है, whereas eukaryotic cell takes at least 24 hours. So it's okay, they have got time to synthesize more copies of polypeptide chain or protein synthesis in slower rate as compared to the bacterial translation rate. ठीक है? Now, so we know that ribosome is composed of large and small subunit. ये हम सबको पता है. and it has got, so your ribosome, the smaller subunit has got your 16S RNA, which goes in binds to the Shine-Dalgarno sequence. If you remember, we have discussed about it, right? So 16S RNA जो है वो आपका Shine-Dalgarno sequence या RBS, that is ribosome binding sequence पे जाके bind करता है. Then comes your 50S subunit, which is recruited with the help of the other proteins. It has got 3 sites: EPA, exit, peptidyl transferase का, and your acceptor or amino cell tRNA के लिए space जो है. ठीक है? So we'll discuss how later how these translation is happening. Okay. So the ribosome is composed of the subunits, large and small. Large subunit contains peptidyl transferase center, which is responsible for the formation of peptide bonds. So guys, please remember, here we are talking about peptidyl transferase. नाम से पता चल रहा है, it has got the enzyme activity, but it is not your protein which is acting as the enzyme. Okay, here it is the RNA which is acting as the enzyme. So what are such RNAs called? Do you remember from the previous lectures? RNA acting as the enzyme, those are known as
Ribosome.
Ribozyme. Yes. So we have discussed during the splicing, right? So even peptidyl transferase is also one of the example of ribozyme. Okay. The small subunit contains a decoding center in which charged tRNAs read or decode the codons units of the mRNA. Okay. Now, this is also something which we already know. The large and small subunits. So in this, that S जो रहता है, हम जो लिखते हैं यहाँ पे 50S, 30S, that S stands for what? So in few cases they say it is a sedimentation quantity or value, and it is named after the Nobel laureate whose name was Theodor Swedberg. Okay, so S is nothing but it's a unit to measure the sedimentation velocity. Swedberg, okay, उसके नाम के बाद से लगा है. So S means अगर जितना ज्यादा आपका value है, okay, it means it is the faster the sedimentation velocity, उतना ज्यादा आपका उसका weight होगा. Okay, larger the molecule उतना weight होगा, इसलिए उसका sedimentation velocity ज्यादा होगा as compared to the smaller subunit. Okay, this point is very important. So higher the sedimentation velocity, larger the your molecule is there. Okay. So in case of bacteria, we know it is 50S subunit and 30S subunit. Together it is 70S ribosome is there. Okay. Now simple. The more expected question is why 50 and 30 is 80, but again the intact ribosome we are getting 70. So what is the answer behind this? It's very common and routinely asked questions. Yes, yes, Akshay, what is the answer? Tanvi, what will be the answer for this?
I don't remember it exactly, ma'am. But 10S gets used up or something like that. I don't remember.
What is it?
The 10S, I mean, the remaining 10 gets used up or something. I don't remember what is it.
So what happens is that, see, when 50 and 30 are coming together, so definitely there are going to be interactions, right? So that interactions make sure that the available water molecules are removed, and that reduces the volume of the entire intact wala. Okay? So because of that, what will happen? The structure becomes more compact, and the additional water molecules जो रहते हैं बीच में, okay, so those are being removed, and hence the total it becomes less as compared to the total, the mathematical total 80 say it becomes 70. So together when it is intact, so it comes to be 70 less. Okay, the space has been removed, the space occupied by the water, it has been removed. And when, see, you know that कोई भी फैली हुई चीज रहेगी, so for them it to settle it will take time, but if you have something very compacted material, so sedimentation rate will be of high for those particular things. It's simple, right? अगर ऐसी कोई फैली हुई चीज है, इसका रोटेट होके नीचे सेटल होना एंड ऐसी चीज का रोटेट होके सेटल होना, किसका सबसे ज्यादा हाई होगा? ऑब्वियसली इसका, राइट? So which has got more compact structure, that will get easily sedimented as compared to this structure. Okay, so that's why उसका जो value है वो आपका कम हो जाता है as compared to the mathematical total value. Okay. So this is just that same explanation. So you are going to have that जितना ज़्यादा आपका नीचे settle होगा उतना ज़्यादा वो larger molecule है. So we found that it is 70 for the intact, larger के लिए 50, smaller के लिए 30. Now in the case of eukaryotes also we know it is your 60, 40 and together it is 80 instead of 100. Okay, so these are the classifications where I mean it is up to you. What I will suggest is as like in the case of the prokaryotes, please do remember that it is 16S rRNA over here and 23S rRNA over here. Okay, 16S 30 का है, 23 50 का है. Similarly 40 का 18 याद रखो और 60 का 28 याद रखो, because question comes based on this. Okay, I'll come to that, like what kind of questions they ask. So as I was saying, so please try to remember this one, this here and here and here. Now why I was saying was because see, whenever you go to any research lab, okay, so if you're working on, let us say bacteria, okay, you're working on any bacteria, like maybe you just went for your MSc project to some lab who are like it's a microbiology lab, and अगर आपको बोला जाए कि like from which place you are and all, so Akshay, you must have heard about the Lonar Lake in Maharashtra. सुना है क्या इसके बारे में?
No.
No, चलो let it be. Tanvi, you must have heard about the let us say that नैनीताल का आप वो कोई भी एक river ले लो, ठीक है? Let us take some river which is like very famous for some reasons, okay? And suppose Tanvi goes to the lab and that scientist says कि आप अपने locality का famous चीज का एक sample लेकर आओ. So Tanvi takes a sample from her favorite place or whatever, snowy area हो या thermo hot springs होते हैं. Like I remember there is one hot spring area in Uttarakhand. Mani something.
Badrinath have it.
Okay, okay. Badrinath is also having. So you know what are hot springs? यहाँ पे आपका खौलता पानी निकलता रहता है, right? So she is taking that sample. Okay. So we now know that there are bacteria which can grow at very low temperature, correct? And there are bacteria which can grow at very high temperature. So of course those are known as saprophiles and these are known as thermophiles, correct? So ये चीज हमें पता है. So these are known as the saprophiles. Okay. So this is something which we already know, but do we know like all the thermophilic bacteria are sequenced? नहीं ना, हमें नहीं पता. Right now people say that इतने सारे bacteria are already identified, but still it is constituting only 0.01% of the total bacterial species present on the earth. So just imagine और कितने बाकी हैं. This is like what? This is nothing, right? और जबकि हमें मालूम है already in lakhs many species are known in the databases. Okay, so for example, she takes this thermophilic bacteria which can grow on the hot conditions. She takes to the lab, and in the lab she maintains that particular temperature, media and everything, and she's growing that bacteria. So first of all, she has to optimize the conditions where she can grow the particular bacteria or the psychrophile. Let us say psychrophile थोड़ा easy रहता है, hot condition maintain करना difficult होता है. Psychrophile का क्या रहता है? You can just put into the cold room temperature, like you can put at the 4 degree या 18 degree में डाल दो, ठीक है? It's easy to maintain. You make a plate of the media, streak कर दो या spread plate कर दो and put into this refrigeration condition, and these bacteria grow very nicely. Okay, but you don't know what is the name or whether this species has been identified before or ये पहली बार है. If it is first time, then you can name this bacteria after your name. Okay, so something like that. Now the first step will be to isolate the bacteria. Of course, isolate the DNA of the bacteria, as already we have discussed in the methods. Okay, remember how, like from the bacteria, how you can isolate and purify the DNA. Now take this DNA. Okay, अब आपका next step क्या होता है? There's something called ribotyping. Okay, ribotyping. What does it mean? It means you are going to use this bacterial DNA, okay, and you're going to identify the prokaryotic ribosomes. Sorry, you're going to identify which DNA or which bacteria it is. तो आप कैसे करोगे? So see, this is 16S rRNA. Okay, it means this sequence is present in DNA also. Do you agree? Yeah, RNA है, right? RNA कहाँ से आया? DNA से आया है. So the sequence which is coding for 16S rRNA, there might be a region or the sequence which is your DNA, correct? ये एक पर्टिकुलर सीक्वेंस होगा जिस डीएनए से आपको ये आरएनए मिला है, करेक्ट? इतना तो मालूम है ना, इतना क्लियर है, राइट? हाँ, तो इस आरएनए का ऊपर डीएनए होगा ही. सो यू आर गोइंग टू आइडेंटिफाई this particular 16S DNA using bacterial DNA. हम लोग बैक्टीरिया का राइबोसोम आरएनए कुछ प्यूरिफाई नहीं करते बिकॉज इट इज वेरी डिफिकल्ट टू गेट द आरएनए. इट्स वेरी अनस्टेबल एज वी हैव ऑलरेडी डिस्कस. ओके, तो द बैक्टीरियल डीएनए लेंगे हम इसका. एंड बैक्टीरियल डीएनए there are universal primers. Okay, so these universal primers are for your 16S DNA. Okay, now these universal primers है, आपके पास bacterial DNA है, you are going to run PCR. Okay, now you are going to get the amplified length of some part of the DNA. Okay, this DNA you are going to give it for the sequencing. Now from the sequencing you are going to get the sequence of the some stretch of DNA. You are going to put into the chromatogram. Okay, get the correct format of the sequence. जो हम FASTA format नहीं बोलते हैं, this is a proper alignment of the sequence, whether it is amino acids or nucleotide sequence, हम FASTA format लेते हैं. Okay, now put this FASTA format into your NCBI. What is NCBI? National Center for Biological Information. ठीक है, अब यहाँ पे भी डाल सकते हो. There is something called BLAST also. अगर आपने बायोइन्फॉर्मेटिक्स में पढ़ा होगा, basic local alignment search tool है it is. ठीक है, so you can put it over there. Now if you get a hit, it means the bacteria was already identified by someone else and it is present in the database. So now you know which kind of bacteria, which species of bacteria is present in your sample. Okay, is this entire experimental procedure is clear?
Yes, ma'am.
हाँ, तो हम ऐसे आइडेंटिफाई करते हैं कोई भी नया बैक्टीरिया हो. ठीक है, अगर इसी तरह से अगर कोई यूकैरियोटिक का करना होगा, सो वी मेक प्राइमर्स अगेंस्ट 18S DNA. ठीक है, हम आरएनए लेते, डीएनए लेते हैं. ओके, सो ये हो गया आपका एक तरीका. नाउ इफ आई डू ब्लास्ट एनसीबी एवरीथिंग एंड आई एम नॉट गेटिंग एनी हिट, okay, it is just saying that it is not present in the database, then it means what? It means then you have identified a new species on earth. Okay, so then you can name that bacteria after your surname or your first name. So you can name that bacteria. Of course, you need to characterize the bacteria, whether it is cocci or bacillus or bacillus tanvi or like that. You can just name it. ऐसा कुछ आप कर सकते हो. Okay, so this is how the importance of 16S rRNA and 18S rRNA comes into the picture. Now, one interview question which we have asked was, you have got the sample, okay, a DNA sample is there and it is unlabeled. So you don't know whether it is prokaryotic or eukaryotic, okay, but your scientist is asking you to prove or give the information whether this DNA sample is prokaryotic or eukaryotic. तो आप क्या करोगे? क्या करना चाहिए? Already discussed. Same approach you need to apply over here. So what are you going to do? Yes, तन्वी, क्या करना होगा? So what you can do is take the DNA, okay, take the universal primer and run. If you get the band, it means it is prokaryotic sample. If you don't get the band, then it is eukaryotic. So with the help of one PCR, you can just give the answer to your guide whether the DNA sample is prokaryotic or eukaryotic. Clear? Simple approach. Okay. I hope it's clear, guys. Okay. Yeah. So now the large and small subunits, they undergo association and dissociation during each cycle of translation. Okay. So this is also known as ribosome cycle. So what we are trying to say over here is that जैसे ही ट्रांसलेशन होता है, ऑफकोर्स एमआरएनए रहा, आपका 30 सबयूनिट आया, 50 सबयूनिट आया, ओके, प्रोटीन सिंथेसाइज हुआ, and again it leaves and it goes and binds to the another mRNA and starts the translation. Okay, so it means what? The larger and the smaller subunit are getting assembled and disassembled during the translation process, and this association and dissociation is known as the ribosome cycle. Simple. So there is a figure over here, you can see. Yeah, so you can see the mRNA is there, start stop is there, first small subunit, small ribosomal subunit comes, then your first initiated tRNA carrying the methionine amino acid comes, then the last subunit comes, then the rest of the amino acid tRNA comes and carry out the translation. So once your polypeptide chain is released, it reaches the stop codon, what will happen? Both the small and the large subunit gets released. Again, these subunits will go and bind to the another mRNA and start the translation. So this entire process is known as the ribosome cycle. Okay. Now let's discuss in brief the translation. So this, what we are going to discuss now is without the proteins, which is helping in the recruitment of those machinery which is required for the translation. हम उसके बिना एक बार डिस्कस करेंगे, ठीक है? तो हमें मालूम है ट्रांसलेशन शुरू करने के लिए you need a small subunit and the tRNA to bind to the mRNA first, ठीक है? तो एक बार ये हो जाता है, then your larger subunit comes and makes your mRNA sandwich between the two subunits. Next is what? It is started. So at the 5' end of the mRNA, the codons are read and the tRNAs are bringing the amino acids. So हर एक बार क्या होगा? Each codon, okay, so one codon means 3 nucleotides. So it is read by the tRNA or decoded by the tRNA. What will happen then? The amino acids are brought and it will be transferred to the peptide transferase where it is going to form the peptide bond. Okay, so ऐसे ही वो separate होता जाएगा, आगे बढ़ता जाएगा. At the end, when it reaches the stop codon, it is going to stop. Okay, and the subunits will get released. And those separated subunits are now available to bind to the new mRNA. Okay, now although a ribosome can synthesize only one polypeptide at a time, but the same mRNA can be translated simultaneously by multiple ribosomes. So it's like, no, I have the figure, I guess. Yeah, so you can see mRNA is over here and एक राइबोसोम है जो रीड करता हुआ आगे चला गया, पीछे से you can have so many other mRNA, so many other ribosomes which are attached. So this entire thing is known as polyribosome. Okay, or it is also known as polysome. So what does it mean? Single mRNA, but multiple ribosomes are attached. So at the end, let us say that here 6 ribosomes are there. So it means at the end of this translation, I'm going to get 6 molecules of the protein. Clear? Is this clear? Polyribosome?
Yes, ma'am.
Simple topic, it's good. So the same thing we have discussed. Yes, huh?
Is the polyribosome condition for overexpression?
Yes, it will result in the overexpression, and this happens in the prokaryotes.
Not in eukaryotes.
Nah. Okay. So, the ability of multiple ribosomes to function on a single mRNA explains the relative limited abundance of the mRNA in the cells. का मतलब क्या? जितना भी RNA हो, उससे ज्यादा आपका प्रोटीन होता है. It's not like अगर एक mRNA है तो एक प्रोटीन. It's not like if it is one mRNA, you can have thousands of copies of your proteins. Okay, it is because of this presence of the multiple ribosomes. Okay. Yeah. Now, this is also important point. So, new amino acids are attached to the carboxyl terminus of the growing peptide chain. So, we know that जब भी कोई भी आपका amino acid peptide bond formation होता है, so it is between the amino terminus and the carboxyl terminus, right? New amino acid जो आएंगे उसके carboxyl terminus में आपका वो attach होगा. Now, if you see the figure, okay, so this is just for the brief translation steps. So you can see it over here. So जो आपका जो purple color में है, that is your like not the purple color, sorry. You can see it over here. So this was your first, okay. उसका amino acid यहाँ पे दे दिया है. And you can see the N terminus of this is free. इसलिए हम कहते हैं polypeptide chain को read कहाँ से करेंगे आप? N terminal to C terminal आप read करोगे. Correct? Correct? Now, whenever new tRNAs are coming, you can see that it is attaching over here. So what is happening? Purple वाला जो है वो blue वाले tRNA के ऊपर आ जाएगा, and this purple will be removed. Okay, then another will come. It will sit on the A side. Fourth वाला जो है अभी fifth वाले के ऊपर जाएगा. So this is how it is getting shifted. Able to follow? Now the question also comes, like whether it is mRNA which is moving or the ribosome which is moving. And why? What is the answer? Like, how do you justify your answer? Question clear hai? See, it is moving. I mean, of course, it has to move because it has to read the codons, correct? So this mRNA is there. आपका ribosome इस तरफ move हो रहा है. But is it the ribosome which is moving in this direction or mRNA is moving in that direction? In either case, the codons are being read. अब आपको ये बताना है which one is moving, is it the mRNA or the ribosomes? Okay, so tell me the answer tomorrow. I'll just stop here. I'll just explain this and we'll stop here. So here the main thing, okay, so you can see that this is your peptidyl tRNA. So peptidyl tRNA means which is sitting at this P side, this one, okay, this one. Now what is happening? You can see already it is carrying the amino acids over here, the amino acids, a long chain of amino acids. Amino acid tRNA means it is a new tRNA which has come to this side. Now what is happening? We can see the bond is formed between this amino and this carboxyl group which is attached over here. So what will happen? A peptide bond is formed over here between the amino group of the incoming nucleotide and the carboxyl group of the previous amino acid. Correct? You can see it over here. So अब क्या हुआ? जो आपका peptidyl tRNA था, that is free now to leave the ribosome, and amino acid tRNA will get shifted to the peptidyl tRNA, and again this process will repeat till the stop codon is being reached. इतना clear है basics of translation? Right. Okay. Now the importance is that can you see the here the structural face of the tRNA, like here it is holding like this and then you have got like this. Can you see? अंतर समझ में आ रहा है? It is carrying the amino acid here and over here. It is not like tRNA ऐसा है और tRNA ऐसा है. एक tRNA आपका ऐसा आता है, दूसरा tRNA आपका ऐसा आता है. Correct? We'll talk about this in the next lecture. So आज के लिए, are you guys comfortable? Like whatever we have discussed, are you okay with it? Thorough with it? All the concepts are clear? Do you feel that confident? Because these all are important for our upcoming lectures. Once we start talking about the proteins and how it is recruiting in the initiation, elongation, and the termination. Okay, so what is important? Like, let us just summarize. So we have discussed about the tRNA, the importance of amino acyl tRNA synthetase, how it is maintaining the correct fidelity of charging the tRNA with its cognate amino acid. Okay, वो कैसे करते हैं वो important है. You should know about the hydrolytic editing editing pocket which is present on the synthetase. Okay. Then of course these are simple ones, ribosome, polyribosome और polyosome के बारे में they have asked in the 2 marks question, इसे मैंने वो mention किया. Okay, and yeah, that's all. Yeah, ये थोड़ा important है, rest all were like for the practical knowledge purpose. Okay guys, so I hope this is clear, believing that. So shall we stop here? Is there any other topic or any question do you have? Yes, Akshay, Tanvi?
Yes, ma'am. I actually have a doubt. Like when I went through the MCQs, so like with the graph, there was a question. So there was a problem that my answer and the answer which was given in the PDF you sent did not match.
Okay. Can you tell me the number of the question? Like it is the same PDF, right? Which I have shared with you.
Yes, ma'am.
Okay.
14 and 15.
Uh-huh.
Question number 14 and 15.
Okay. Okay. So I'll just go through that question and maybe in tomorrow's lecture I'll include and we can discuss then tomorrow. That
Okay.
sounds fine. Yeah. 14th and 15th, right?
Yes, ma'am.
15, right? Is it in the continuous or is it section wise? I believe it is in section wise, but the number is in continuous number.
Yes, ma'am.
Okay, okay. I think it is from what, replication, transcription?
A replication.
Okay, okay. No problem. We'll discuss. No problem. Okay then. Okay. Chalo then. Bye guys. Good night.
Not, ma'am.
Thank you, ma'am.